Have you ever wondered how tiny microbes, too small to be seen with the naked eyes, manage to survive in diverse environmental conditions? Have you ever wondered how they manage to survive in boiling hot spring, deep in the ocean, or even inside our body? The answer to this lies in nutrition. Just like us humans, microbes also need food and nutrients for their growth and survival. They have different ways to obtain these nutrients, but their strategy to obtain these nutrients are far more diverse. They're very creative and sometimes very dangerous. In fact, the very nutrients that microbes use can determine whether they live harmlessly or they can cause deadly infection in humans. So in this micro lecture, we are going to dive deep into our chapter 9, which is microbial nutrition. We will look at what nutrients microbe needs to survive, how they obtain them, and why this is really important for medicine, for our ecology, and also for application in biotechnology. So we'll really pay attention to each of the nutritional components that helps microbe to survive in the environment. But let's start from the basics and understand nutrition first. So nutrition is the process by which microbes can obtain nutrients from the environment. Now, these nutrients are able to provide various supports, energy supports, metabolism for the microbe. The microbe also uses these nutrients to perform most of its cellular activities. These nutrients are obtained in chemical forms, and they come in variations depending on the type of microorganisms. So how much of an element is needed by E. coli may vary from how much may be needed by Staphylococcus aureus. Now, there are various elemental nutrients such as carbon, hydrogen, manganese, chlorine, among several others. And these are required to help in the functioning of the microbes. Now we'll get to look at the cellular composition of microbes, the major macronutrients in microbes, and why these elemental forms are really required to help sustain microbes. Let's look at the term essential nutrients. These are substances or nutrients that must be provided to the organism, mainly because the cell cannot synthesize it themselves. There are 6 major essential nutrients, which I call the CHNOPS. So these are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. The essential nutrients can be categorized into 2. Macronutrients, which are needed in large quantities, and micronutrients, also called trace elements, which are required in smaller quantities. Macronutrients include the CHNOPS. So carbon is a macronutrient, hydrogen is a macronutrient, and they play an important role in cell structure and metabolism. Micronutrients, on the other hand, such as manganese, iron, nickel, although needed in smaller quantities, they play an important functional role in enzyme activity as well as maintaining the protein structure. Now, microbes can obtain these essential nutrients, whether macro or micro, from inorganic sources or organic nutrient sources. And there are various components to this. So carbon, oxygen, nitrogen, all these different elemental sources can be obtained from inorganic environmental reservoirs. So for example, carbon can be obtained from the air through carbon dioxide, from rocks, from sediment. Hydrogen can be obtained through water or hydrogen gas. Now, if you look at these elemental nutritional requirements, it is also important to look at the chemical composition of a bacterial cell. Now, the chemical composition highlights the nutritional requirement and why these organic compounds and the essential element are really key for the bacterial survival. Now, a typical bacterial cell is composed of 70% water in its cell content. Now, if you have the dry cell weight, about 96% of the dry cell weight is composed of the CHNOPS, which is carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And this kind of highlights why these elements are needed and must be available for use to help support the cellular composition of these microbes. So a little bit more detail. If you take, for example, a microbe like Escherichia coli or E. coli, it is made up of these major organic compounds. Protein is about 50% based on the dry weight. Carbohydrates and lipids are about 10% each. When you break it into the elemental form, carbon is about 50% of the dry weight. Oxygen is about 20%. So the elemental component of the cell highlight why these elemental nutrients are required for the functioning and survival of these microbes. Now let's pause and do a quick micro check. If you take an element like iron, which category do you think they belongs to? Macro or micro? Feel free to pause and share your answer with me in the comment section. So in our video lecture, we would be focusing on these 6 key CHNOPS, essential macronutrients. We'll look at their importance to the cell, and we'll look at how microbes obtain them. We'll also highlight if any of these nutritional composition has been associated with medical relevance or a disease condition. Let's start first with carbon. Microbes can obtain carbon from inorganic source or organic source. Now, if an organism obtains carbon from organic source, they are called heterotroph. A typical example of a heterotroph is Pseudomonas aeruginosa, which thrives in the lung area. Various Pseudomonas species are heterotroph. They can obtain carbon from 100 different carbon substrate sources. Now, microbes can also obtain carbon from inorganic carbon dioxide. These are called autotrophs. Auto means self. Troph means feeder. So self-feeders are able to obtain their carbon from carbon dioxide, and they can convert it into various carbon compounds through the process of photosynthesis. A typical example of autotroph is cyanobacteria. Cyanobacteria is a photosynthetic bacteria. It obtains its carbon from carbon dioxide, and it can make its own food through the process of photosynthesis. So Pseudomonas aeruginosa is an example of a heterotroph, and an example of an autotroph would be cyanobacteria. Now, the second element we'll be looking at is nitrogen. Now, majority of the nitrogen reservoir exists as nitrogen gas. That is about 79% of the atmosphere. Now, nitrogen is an indispensable part of major macromolecules in the cell of microbes such as proteins, nucleic acids such as DNA and RNA, and even ATP. Now, since nitrogen exists as nitrogen gas, this form cannot be fully utilized by most microbes and most organisms. But there are some bacteria and some algae which are able to process this into more usable form, such as nitrate and nitrite, that can be used and absorbed by other microbes. So, for example, if you take some bacterial species, they are able to fix nitrogen and process it into a more usable ammonia form. So nitrogen must be converted to ammonia before it can enter the cell, and through the process of nitrogen fixation, these nutrients can be more accessible to microbes. Now, when you look at these two elements that we've talked about, some of these nutrients have been associated with diseases. So, for example, in the case of carbon, Pseudomonas aeruginosa species have been associated or linked with 90% of cystic fibrosis death. Now, cystic fibrosis, you have the bacteria thriving in the lung area, and 90% of cystic fibrosis dead cases have been associated with this particular bacteria. So whether carbon or hydrogen, its source and its availability is really key, as there are medical relevance to that. The third nutrient we'll look at is oxygen. Oxygen. Oxygen can be obtained through water. It can also be obtained through oxygen gas. Oxygen is crucial for respiration, for enzyme function, and for cell structure. Some microbes are able to thrive on oxygen gas, and these are called aerobes, while others are killed by oxygen, which are anaerobes. So anaerobes, oxygen is poisonous to them, and we'll discuss in some of our upcoming chapters why oxygen is poisonous to anaerobes. Now, a common form of inorganic source is oxygen, and oxygen gas makes up about 20% of the atmosphere. Our fourth nutrient in the macromolecules we are looking at is hydrogen. Hydrogen can be obtained by microbes through hydrogen gas, through water, and hydrogen has three important roles in the cell. First, it helps to maintain pH balance and is also important in the transfer of energy, especially during redox reaction, as it can serve as a source of free energy, and is also important in stabilizing molecules in the cell through creating hydrogen bond situation. The fifth element source is phosphorus or phosphates. Now, phosphorus is the backbone of nucleic acids such as DNA and RNA, and they are even part of ATP as well as phospholipid in the cell membrane. Without phosphate, the cells cannot reproduce. So phosphorus or phosphate is a really key component of nucleic acid and is very essential for the genetic of cell and especially for viruses. Now, the sixth and last nutritional source is sulfur. Sulfur is found in some amino acids such as cysteine and methionine, and sulfur can be obtained through rocks and sediment. They are widely distributed in the environment. Sulfur is important in creating disulfide bonds, which helps to give the three-dimensional shape and structural stability of proteins. Now, let's also look at some other factors and sources that are not part of the CHNOPS or the 6 essential macronutrients, but they are equally also important for the microbial cell. The first we'll look at is growth factors. So these are organic compounds that cannot be synthesized by the cell. So they must be provided from the environment. Examples of growth factors include vitamins such as nicotinamide adenine dinucleotide, amino acids. These must be provided through the environment for the microbial cell to utilize because they cannot be synthesized by the body. Microbes also need energy, and they can obtain energy from various sources. So we're going to look at different energy sources classification and how microbes are able to obtain energy through these sources. The first that we'll look at is photoautotrophs. These are microbes that use lights and can obtain their energy through the sunlight. Typical example is cyanobacteria as well as algae. So photoautotroph can use the sunlight's energy source and carbon dioxide to synthesize their own food. The second energy classification would be chemoautotrophs. Chemoautotrophs. So these can oxidize inorganic compounds, and this is very typical for nitrifying bacteria. You can also have chemotrophs. So these are microbes that can gain their energy from chemical compounds. So you either obtain energy through the sunlight as a phototroph, or you can generate your energy through chemical compounds. So we've also mentioned photoautotroph, they use sunlight, and they can also use carbon dioxide to make their own food. So typical example, cyanobacteria is both a phototroph and is also an autotroph. So it's a photoautotroph. Now you can also have chemoorganic autotroph. These use organic compounds for their energy, and they use inorganic compound as their carbon source. So they can use inorganic from carbon dioxide as their carbon energy source. Chemolethal autotroph. These are organisms that they don't need sunlight nor organic nutrients. They can rely solely on inorganic material for their energy and for their carbon source. And they are able to remove electrons from these inorganic substrate or materials to be able to get their energy component as needed. A typical example of a chemoheterotroph would be E. coli. So E. coli is a chemoheterotroph. Now you can also have methanogens. Methanogens. So this belongs to the archaea group, and they're able to produce methane gas from carbon dioxide and hydrogen gas. Now, chemoheterotrophs, they can obtain both their carbon and energy source from organic compound. And Escherichia coli, fungi, these are typical examples of chemoheterotroph microbes. Now let's look at some other special nutritional types that are very unique for some microbes. So we'll look at two classes, Saprobes and Halobacteria. So Saprobes, they live on decaying organic material. So for example, fungi is a saprobe. It can decay leaf litter or materials. And we can also have halobacteria. So these are halophiles, and they can survive and thrive on salty environments, such as in the sea. So saprobes, they are decomposers, and they are very essential for recycling nutrients. So as they perform their decomposition activity, they help recycle nutrients back into the ecosystem. Mostly bacteria and fungi are saprobes. Now, saprobes are able to release enzymes into the environment to help digest these recycling nutrients and materials or particles, and then they transport it into the cells. They digest it on the outside, and then they take the byproduct of their digestion into their cell. So typically, if you have a bacteria or a fungi, which is a saprobe, and there's a decaying organic debris, they are able to release enzymes onto this debris on the outside to digest it. And the enzymes are able to break down the nutritional component in this organic debris or waste and break down all the bonds to release the needed nutrients and molecules in them. Now we also have obligate saprobes. That's another key term I want you to pay attention to. These are microbes that exist strictly on dead organic material. So protozoa, fungi, and bacteria, these are typical obligate saprobes that thrive on organic matter either in the soil or in water. You can also have facultative parasites. These can infect living hosts. If given any chance, they would come in, take over the cell and the machinery of the host when the host is compromised and cause infection to the host. Now, typically for microbes, when they obtain these nutrient sources, how do they transport them into the cell to be utilized? So we're going to look at 5 different transport mechanisms by which microbes are able to transport their needed nutrients into the soil, into their cell. The first one we'll look at is diffusion. Diffusion is the movement of molecules from a higher concentration to a lower concentration. Now, movement of molecules in the process of diffusion is a passive process. You do not need any form of energy to move nutrients through the process of diffusion. So example, oxygen gas, carbon dioxide, they can easily be diffused into the cell through the process of diffusion. Now let's look at how this video demonstrates the process of diffusion.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When of their lower concentration, molecules, and the steepness of the...
So in this video, we can clearly see that when you put in the sugar molecules, it's going to be in an area of higher concentration, and it begins to diffuse and spread across to be evenly distributed until there's an equilibrium throughout the entire solution.
Molecules.
Now, the second nutrient transport mechanism is osmosis. Osmosis is the diffusion of water through a selectively permeable membrane. Now, osmosis reflects the selective permeability of the cell membrane, even as it transports water movement in and out of the cell. Now, depending on the condition of the cell, either an isotonic environment, a hypotonic, or a hypertonic environment, the movement of water would be different. In the case of osmosis, water will be moving from where there's high water concentration to where there's low water concentration. Now, if you take an isotonic solution, this is a solution which is a balanced solution. So a cell in such environment, you would have net movement of water. The amount of water coming in would be the same as the amount of water coming out. So how water moves through the process of osmosis would depend on the solution that the cell finds itself in and the type of cell. Now, in the case of eukaryotic cell, like we find for humans, there's no cell wall, there's a cell membrane. So in isotonic solution, the amount of water gain would be the same as the amount of water which is lost. And there's going to be a net water movement. In the case of hypotonic, more water would be moving into the cell, And because there's no cell wall, the cell cannot survive and is going to lead to cell lysis or burst. But in situations where the cell do have a cell wall, the situation would be different. So let's take a look at various conditions. If you have a microbe like amoeba, which is a protozoa, they usually have contractile vacuoles, so they might be absorbing water in a hypotonic environment, but they are still able to survive because their contractile vacuoles allows them to take in all these excess water and push it out of the cell. Bacteria also have cell wall, and in a hypotonic environment, they might absorb so much water, water will be entering the cell, but the cell would be protected from bursting because of the cell wall. Now, in a hypertonic environment, cell is going to lose water. So either there's a high salty or salinity environment, a high sugary environment, the cell is going to lose water to the environment. So in this table, let's look at a summary of how movement of water happen for animal cell compared to a bacterial cell in either of these solution type. So in an isotonic, where you have same amount of solute and solvent, there's always going to be a net movement of water, where the cell is going to remain stable. In a hypotonic, because you have less solute, water is always going to enter the cell. For animal cells, the cell is going to burst because there's no cell wall. For bacterial cell, water is going to enter, but the cell wall is going to protect the cells and prevent the cell from being lysis or breaking. In hypertonic environments, where you have high solute concentration, water was going to leave the cell in the case of animal cells, and the cell would begin to shrink or become dehydrated. So for example, if you put a cell in honey or a high corn syrup or fructose solution, these are all hypertonic solutions, so the cell is going to become dehydrated. In the case of bacterial cell in a hypertonic, the cell will lose water, but instead of the cell shrinking, it will go through what we call plasmolysis, where the cell walls separate from the cell membrane and the cytoplasm also shrinks. Now let's have a second micro check question. If you have a bacteria which is in the Dead Sea, which is very salty, removed and placed in a freshwater environment, what do you think would happen? Based on what we've discussed for osmosis and different solution and conditioning, if you move a bacteria from a Dead Sea environment, which is mainly salty, hypertonic, and you place it in a freshwater environment, what would happen? Please share with me your thoughts in the comment section. Now we are still talking about other nutrient transport mechanisms. So we've already mentioned simple diffusion, where there's movement of molecules. There's also a type of diffusion called facilitated diffusion. Now with facilitated diffusion, there are various membrane proteins which help to carry specific molecules that bind to them through the cell membrane and transport these molecules into the cell for it to be used. Now, for facilitated diffusion, there are carrier proteins. They take the molecules, bind to it, and take it across in and out of the cell membrane.
Known as
Active transport is also another transport mechanism. Now, with active transport, you are moving nutrients against their gradients. So active transport is like you are climbing a hill. So you are pushing more nutrients towards where there's already more nutrients. Now, you would need the help of membrane proteins, mainly permeases and pumps. Now, these are going to act as carriers with a lot of energy required to help push nutrients towards where there's already more. Now, a typical example of active transport is sodium-potassium pump in humans, where sodium is moved out of the cell and potassium is moved into the cell. Now, the sodium-potassium pump is one of the commonest pump where the pump has a location to pick 3 sodium to move it outside to the cell of the cell, and it has a location for 2 potassium where it takes on 2 potassium and move it into the cell. But this, because you are moving against the diffusion gradient, you need energy to be able to perform this. Now, a typical example of an active transport mechanism is group translocation, and group translocation is mainly used by some bacterial species. With group translocation, you are moving a molecule across the membrane, but you simultaneously convert it to a metabolic useful form. So, for example, a bacteria may be moving molecules through group translocation, but as they move it, they can convert it to a metabolically phosphorylated form so that it can become more useful and it can stay inside of the cell for them to be able to use. Now, sometimes nutrients need to be transported in bulk amounts. So when we need mass transport of large particles into the cell, bulk transport mechanisms has to be done. And this is called endocytosis. There are 2 categories of endocytosis. Phagocytosis, where you are moving bulk solid particles. And the cell is always going to use vesicles to be able to move these bulk particles into the cell. Pinocytosis is when we are moving bulky lipids. It also uses vesicles which engulf around the liquid and help move it into the cell to be able to use. Now, phagocytosis is typically used by amoeba, which is a protozoa, and is also the mechanism by which our white blood cell type perform its immune protective function, where our white blood cells are able to ingest whole viral or bacterial particles through phagocytosis, engulf it, bring it into the cell to be digested or to be degraded. Now, with pinocytosis, we can move liquids like oil and other needed molecules into the cell. So let's have a quick recap. Just like humans, microbes also need nutrients to survive. They can obtain their nutrients from different sources. It can be organic sources, it can be inorganic sources. But these 6 nutrients are essential, the CHNOPS, because they are key components of the chemical composition of these microbes: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Now, microbes can obtain these either carbon source, hydrogen source, energy source from organic or inorganic sources. And we also looked at various categories that they can obtain these nutrients as well. Now, when they obtain these nutrients, microbes have different transport mechanisms such as diffusion, osmosis, active transport, or endocytosis, by which they transport these to be utilized inside of the cell. Let me know if you have any question. But I want you to think about this video and give me one word wrap. What is one main thing that caught your attention as you listen to this video? Share with me your one word wrap in the comment section. Thank you.